Hello, and thank you for visiting my poster. I'm sorry I couldn't be there in person, but I'm happy to share my work with you here virtually. My name is Haley Dimon, and I'm a PhD student in neuroscience who studies under the supervision of Dr. Melissa Henry, who does have the pleasure of being there with you today. This poster is highlighting some preliminary data from a pilot study that aimed to determine the most appropriate primer set to use for a 16S sequencing experiment of the gut microbiome. This project is part of a larger prospective longitudinal study aimed at identifying biological, psychological, and social risk factors for pain and distress in people newly diagnosed with cancer. If we're all at the Psycho-Social Oncology conference, I imagine we all appreciate the suffering and distress a new diagnosis of cancer can have on a person's life, and how this can subsequently compromise quality of life, treatment adherence, and even survival. Here we are aiming to recruit 150 people newly diagnosed with cancer of the breast, lung, or head and neck, as these three sites are associated with elevated levels of distress and pain. We then asked individuals to provide a stool sample at baseline and complete a series of validated questionnaires that assess sociodemographics, mental health, health literacy, childhood abuse, stress, and quality of life. These questionnaires are repeated every 3 months for a year. We also conduct structured clinical interviews for DSM-5 diagnoses. Between the methodology and the objectives, you can see the recruitment progress as of a few months ago when this poster was created. We have about 75 individuals who have consented, 50 of whom have then completed baseline questionnaires for a participation rate of about 67%. As I have mentioned, this poster highlights the data from a small pilot study done with the first 25 stool samples from study participants and 5 healthy controls. These stool samples were then sequenced using 16S sequencing. This approach involves the sequencing of a specific region in the 16S ribosomal RNA gene, which is present in all bacteria. So this gene contains both conserved regions present in all bacteria and variable regions, which vary between different species and can help us identify them. So to initiate the sequencing process, we use a short DNA fragment known as primers. Here we are comparing a V4 primer and a V5-V6 primer. So these primers are designed to target and copy variable region 4 and variable region 5-6 respectively. By examining the variations in these sequences, we classify bacteria into different taxa down to the species level by comparing the data to data libraries of bacterial sequences. First, we wanted to see how well the primers could explain the bacterial diversity, which can be seen in figure A and B. Both sets of primers had similar diversity at the phylum level, represented by color in the bubble plot, where the different species within a phylum are represented by circles, whose sizes represent relative abundance. In the bottom of panel A, we see that both primer sets have good resolution, that species annotated sequences account for almost 6 million counts using the V5-V6 primer. The V4 primer was able to detect 68% of the total abundance, whereas the V5-V6 primer detected 76% of the total abundance. In panel B, we see a representation of the alpha and beta diversity profiles from the V5-V6 primers. Alpha diversity centers on the intricacies within an individual sample, quantifying both species richness and evenness. Beta diversity interrogates dissimilarities in species across multiple samples. It evaluates the dissimilarity of microbial communities among diverse samples, and then we can decipher patterns of similarity and divergence across distinct experimental groups. Here, as this was a pilot study, we wanted to probe our sample with a simple question and minimize confounds as much as possible to have a clear answer. So here we just simply compared healthy controls to study participants. On the left, we can see the alpha diversity value for healthy controls is significantly higher than our patient population, suggesting that cancer reduces bacterial richness and evenness. Of course, the ulterior explanation could be that this reduction in alpha diversity predisposes one to this illness. The V5-V6 primer shown here had a p-value of less than 0.001, whereas the V4 primer was still able to detect a significant difference between groups, but with a p-value of less than 0.01. Similarly, both primer sets were able to detect a significant difference between patients and healthy controls' beta diversity, using both a constrained and unconstrained model of assessment, and both primers had a significant difference, p-value of less than 0.01. We then were curious to see if the bacterial data separated on psychosocial dimensions, and we were happy to see that even in this small pilot study that indeed yes. So in panel C on the top, the red points represent study participants with cancer who at baseline had anxiety, and this was detected with the hospital anxiety and depression scale. The points in black are those individuals who do have a newly diagnosed cancer but do not have anxiety as detected by the HADS. We see a clear separation of bacterial profiles between those two groups. And when looking at the species present in these two groups, individuals without anxiety had higher levels of commensal bacteria, and individuals with anxiety had elevated levels of bacterial species associated with anxiety and other mental health pathologies in the literature. So this suggests that at baseline, bacterial profiles could indicate who is predisposed to anxiety during a cancer trajectory. Next, instead of separating participants based on psychosocial dimensions, we used a clustering algorithm to cluster bacterial profiles, and it yielded 3 cluster solutions. So I'm not going to go through every single difference in species in each cluster. I just wanted to highlight a few key ones. In cluster 1, there were elevated levels of the Lactoclostridium schneidin, which is associated with cancer and pain in the literature. Cluster 2 had elevated levels of Lactnospira elegans, associated with breast cancer and stress. Cluster 3 had reduced levels of the Mediterranobacter glycyrzinlictis bacteria, which is associated with inflammation and obesity. Of course, we can't draw any conclusions at this point in time about which species play what role. However, it does suggest that we would be able to repeat this approach at the end of our experiment and see how bacterial cluster membership is associated with pain and distress outcomes. To conclude, our pilot study showed promising results that distinct groups of individuals had distinct gut microbiome profiles, and that our sequencing and analysis approach is sound. Once all of our samples are collected, we will move forward only using the V5-V6 primer, as it provided greater resolution and more bacterial diversity detection. We hope to use the gut microbiome as a novel biomarker for distress and pain, particularly in an oncology context. But also, it could guide novel approaches for pain and distress management by regulating the gut-brain axis. With that, I would like to thank you for your attention and coming to visit my poster, and I'm happy to answer any questions by email. You can find all my contact information on the same site where you found this video. Again, thank you for your attention and enjoy the rest of IPOS.